Hello and welcome to my channel. This video is how my PhD in pharmacology and all the experiments and papers and articles that I published that I had to do while obtaining my PhD in pharmacology has led me to have an understanding for the mechanism of emergence. So these are the four main publications that I conducted, wrote, and published, and you can see that two of them are about breast cancer. One of them is about the origin of life and evolution, and another one was published in the first edition Taylor and Francis CRC Press book in the quantum mechanics of biological systems. So the quantum mechanics of biological systems, the origin of life, and metastatic breast cancer might not seem to be things that are related. However, they're related due to biomolecular condensates. And biomolecular condensates are how biological molecules, for instance, proteins, ions, sugars, lipids, how they all come together on the microscopic scale, smaller than a cell, and how that can form all the different morphologies that we see in cells and tissues. So understanding how that forms will help you understand how all these other emergent geometries form. So to continue, we'll just look at a summary of what we're going to talk about. First, I'll begin with the first hypothesis that I had in lab and how that led to all my other experiments. So how this hypothesis directed my research into biomolecular condensates and liquid-liquid phase separation. Liquid-liquid phase separation is how biomolecular condensates form and coalesce. Then I'm going to go through each question that I asked and how that led to the experiments that I conducted, and how what I learned enabled me to write an article about the mechanism for emergence via liquid-liquid phase separation in the book titled Quantum Mechanics of Biological Systems that you saw in the previous slide. And then I'll also end with some preliminary experiments of how I started to test this mechanism and how I think we can go about testing it in the future. So the first question that I asked was, how can tocotrienols interact with so many molecular pathways and disease states? So there's two forms of vitamin E, tocopherols and tocotrienols. Tocotrienols is what the lab that I started doing research in to obtain my PhD was looking at and had been looking at for years. There's a many labs that have published papers on tocotrienols and how they have anti-cancer effects along with other effects in many different disease states. So they can be used to prevent cancer, maybe as a cancer treatment in the future, and they've also been shown to be effective in cardiovascular disease, in neurological diseases, and in skin diseases. And I would like to mention there's 8 different forms of vitamin E, so 4 different tocopherols and 4 different tocotrienols, and each form is called alpha, beta, gamma, delta, tocotrienol, and tocopherol. However, if you are using tocotrienols, you want to make sure not to use any tocopherols because the tocopherols will inhibit the absorption of the tocotrienols. And specifically, my lab was looking at gamma tocotrienols because it was the most bioavailable. However, delta tocotrienol is the most potent form, and both delta and gamma tocotrienols can be found in the annatto plant, which doesn't have any tocopherols. So it's, if you're just looking for tocotrienols, that's the best source. So in trying to answer the question about what exactly does tocotrienol bind to that can explain all its effects, I looked into galectin-3, which is a beta-galactoside and farnesyl binding protein. So it has a dual mode binding site. It has the carbohydrate recognition domain, which can bind sugars like beta-lactose, and it also has a hydrophobic binding pocket, right under the carbohydrate recognition domain. So the hydrophobic bonding pocket would be about right here, and the carbohydrate recognition domain is binding beta lactose, shown in pink right here. And what's not shown here, which would be on the N-terminal tail right here, is the intrinsically disordered tail, which is about 46% of the total length of the protein. So the reason why it has this dual mode binding site is so that it can act as a framework protein. So it's similar to a structural protein, but it's not considered a structural protein because it doesn't aid in any mechanical stability like collagen fibers or fibronectin fibers, but it does help with context dependent signaling because it forms a dynamic lattice that allows it to move around quickly, to form and dissolve, which helps other proteins stay in the context they need to be, the subcellular context that they need to be active. For instance, it can hold the ROS protein on the inner leaflet of the plasma membrane, and that's where ROS needs to be active. And the way it does that is because ROS has a post-translational modification, and you can see that this phernosyl group looks very similar to tocotrienol, and that's what gave me my first idea that tocotrienols might work on galectin-3. So when ROS has this phernosyl residue, it can bind to galectin-3 and be held in place. So that's just one of many examples of how galectin-3 can can behave and function in a context-dependent manner inside the cell to shuttle proteins and hold proteins where they need to be. So another way that this happens is outside the cell, specifically through branched in glycans. So it's branched in glycans or branch sugar residues, and beta lactose is normally used as a competitive inhibitor to inhibit the binding of these branch sugars. But how these branch sugars function normally is they are attached to glycoproteins, for instance, tyrosine kinases and integrin receptors outside the cell. And then those receptors will bind the galectin-3 lattice, and then the galectin-3 lattice will stabilize those receptors so they can function in terms of the integrin receptor. They would function for adhesion formation, focal and fibular adhesion formation, where those adhesions would be attached to collagen or fibronectin fibrils. So the galectin-3 would help in a dynamic way stabilize those receptors connecting to these fibers outside of the cell. So to continue, how exactly does galectin-3 function? I explained to you the protein mechanics, but how does it function with other proteins and how does it form this lattice that I mentioned? It oligomerizes, which just means it binds to itself to form a dynamic lattice via liquid-liquid phase separation. So it can form dimers, trimers, tetramer, and pentamer. And these are just the basic subunits for the lattice that it can form. So it's a three-dimensional lattice. And the way this three-dimensional lattice forms is by, again, binding to post-translational modifications on other proteins like phernosyl residues and branched sugars through glycosylation. And whenever you have, for instance, enough branch sugars outside the cell, it will cause the galectin-3 protein to oligomerize, and then that will begin the lattice formation. And the lattice formation is held together by liquid-liquid phase separation, which just means that the water is in a different phase than the dilute phase of water surrounding it, so it compartmentalizes these protein complexes. So one way you can think about this function of water and this ability of water to be able to do this is by thinking about prime numbers and how they follow hidden patterns without simply repeating. So the question is, does water organize itself using similar hidden non-repeating patterns? Because prime numbers can form symmetrical or non-symmetrical patterns, just as water can form symmetrical dodecahedrons or irregular pentagonal structures. So just in prime numbers, so just talking about math here, inside math or hitting patterns that have you can see you can look on the internet and find papers that prime numbers have some of the same structures as quasi crystals or fractals. So inside math you have these hidden patterns, and that's what I think is basically the one of the main mechanisms or attractors for emergence, is because you'll see that these patterns will pop up again and again in nature, because nature is just organizing based off these fundamental principles of math. So one way that happens is even in liquid water, you have different structures that are constantly forming and getting dilute again. So the structures that form in water are called low density state structures, and you can see that it forms these polygonal structures, and they're low density because you obviously have more space in between the water molecules here. And liquid water is constantly fluctuating from this state to a high density state where you don't have as much space in between the water molecules, and the water molecules are all um, kind of crushed together and randomly uh, mixing together compared to the low density state. So the low density state is what I think the proteins, the DNA, and the RNA that function through liquid-liquid phase separation, that's what I think these proteins stabilize, is this low density state. And this low density state acts as a basically a water framework or a water scaffolding to hold these proteins dynamically together. And this is useful because, just like I said previously, you'll have the galectin-3 protein binding to many different proteins. You'll have it binding to proteins in the extracellular matrix like collagen and fibronectin and laminin, and you'll also have galectin-3 binding to receptors on the membrane. So it's coordinating these huge protein complexes and incorporating different electrical, mechanical, and chemical signaling into these huge protein complexes. So it helps that this organization of water is constantly going through high and low density states, because that will continually reorganize the intrinsically disordered portions of the galectin-3 lattice. So you'll have the galectin-3 lattice incorporating all these different proteins and ions and uh, receptors on the lipid membrane and lipids even. And these high and low density states of water will constantly be reorganizing and shuffling the different conformations of each galectin protein in the lattice, so that it can incorporate all the different signals, electrical, mechanical, and chemical signals, into the totality of all the protein complexes that are trying to combine together. So this is a very interesting way of how these small protein complexes can form so many different structures. And you can imagine that since this happens inside the nucleus, in all organelles, in the cytoplasm, and outside the cell. This is what mediates cell morphology and all the different tissue patterns you see in multicellular life today. So to continue, an easy way to think about liquid-liquid phase separation is oil and water. You can think of proteins that are organizing in biomolecular condensates, will form little bitty droplets in water, just like oil forms little droplets in water. However, biomolecular condensates or water droplets in water. And the way I would describe it is low density water droplets in a high density aqueous water environment. So the way that that happens, how do all of these proteins and waters form these low density networks, is through hydrogen bonding, electrostatic bonding, and hydrophobic bonding. So hydrogen bond, and these are all intermolecular bonding. So hydrogen bonding can happen between proteins and other proteins, and also between proteins and other water molecules and water molecules and other water molecules. Electrostatic bonding can happen between ions and water molecules and ions and the proteins. And a good example of this is sodium chloride has been shown to regulate liquid-liquid phase separation. And you also have hydrophobic bonding, which is how the interior of proteins is held together. So all these three different intermolecular forces is what holds together and dynamically holds together the structure of the biomolecular condensate. And you can see that galectin-3 functions as a biological amphiphile by having a hydrophilic head, mainly hydrophilic, except for the hydrophobic bonding pocket right here. All the all the um, all the amino acids that are exposed on the C-terminal region are all hydrophilic, and that's so it can bind to the sugar residues and remain dynamic, while the N-terminal portion of this protein is mainly hydrophobic, which allows it to bind to other hydrophobic tails so that it can oligomerize and form these lattices that liquid-liquid phase separate. So the way this works, if you want to put it on a graph, is you need the right concentration of proteins, ions, different post-translational modifications like the branched in glycans, the sugars. And whenever you have a high enough concentration and it passes a critical threshold, it it forms these droplets and it forms a condensed phase. However, if some of those proteins or some of those sugars get degraded and it drops below a critical threshold, then that condensate will dissolve back in the water and go into a dispersed phase. So a good cellular signaling mechanism that utilizes liquid-liquid phase separation is the Wnt signaling, signal transduction pathway. So whenever Wnt isn't bound to the membrane receptors, you have liquid-liquid phase separation of these proteins here, including beta-catenin, and beta-catenin will be phosphorylated, which will initiate its degradation. So that's when the Wnt pathway is inactive. But when Wnt is bound to the receptor proteins, then that allows beta-catenin to go freely into the nucleus and organize through liquid-liquid phase separation with other transcription factors so that the beta-catenin targeted genes will be activated and so that'll be the active Wnt signaling pathway. So that's just one of many different examples of cell signaling mechanisms that are regulated by liquid-liquid phase separation. However, they also, liquid-liquid phase separated biomolecular condensates can also function to determine cell morphology. So we'll talk about that more in the future slides, but this is just a rough um, figure right here showing that depending on all the different interactions of the biomolecular condensates can lead to a spherical or epithelial morphology or an elongated or mesenchymal morphology. And a good protein that has been shown to do this is galectin-3, which has been shown to mediate the epithelial to mesenchymal transition. So in epithelial cells, galectin-3 is found inside the cell, inside the cytoplasm specifically, and in mesenchymal cells, it's found outside the cell and inside the nucleus. So that's one good example of how galectin-3 can and how biomolecular condensates can mediate cell morphology and signaling mechanisms. So how can this be used to understand cancer? And we've already talked about the mesenchymal transition, the epithelial to mesenchymal transition. So this is just showing what we previously talked about. When galectin-3 is outside the cell, it's bound to integrin receptors and fibronectin fibrils. And the fibronectin fibrils bound to the integrin receptors, beta 1 integrin receptor shown here, then that will form fibrillar adhesions, and fibrillar adhesions align with the intracellular cytoskeleton. So inside and outside the cell, the actin stress fibers and the fibronectin fibrils are aligning together to form these cell protrusions so that the mesenchymal morphology has the familiar elongated morphology with protrusions. So I did these experiments to show how this is happening. So in the control cells, we have highly malignant MDA and B231 breast cancer cells, and one sign of highly malignant breast cancer cells is when you have a syncytial polynucleated cell. So it's syncytial because it's multiple nuclei surrounded by an actin cytoskeleton shown in red here. And the DNA is stained with a DAPI counterstain, and the adhesions stained green right here, which you can see are elongated in the control cells and are at the tips of the actin stress fibers that is stained with vinculin. So, and vinculin is part of the intracellular adhesion plaque for focal adhesions. So the way that I initiated the generation of these multinucleated cells is by starving them of serum. So I was growing them in 10% growth media, and then in their treatment media, the defined media, I used low amounts of epidermal growth factor. So they went from serum to just having epidermal growth factor. So they were serum starved, and so that stresses out the cells and initiates cell fusion and polyploidal giant cancer cell generation, which you can see in this picture. So I also wanted to show that this mechanism could be stimulated another way, and I further stressed out the cells with 200 millimolars of sucrose. So this 200 millimolars of sucrose will decrease the glycolytic flux inside the cell because this condition of 200 millimolar of sucrose causes hyperosmolarity. So since that decreases the glycolytic flux, you'll have the hexosamine biosynthetic pathway, which is integrated through galectin-3, and it acts as an on and off switch. Whenever you have enough sugar, you have a lot of branched and glycans being formed, and you have glycolysis active. So whenever a lot of sugar is available, you'll have glycolysis active, and that will allow a lot of sugars, branch sugars, in the periphery of the cells. So that's why in these cells you'll see elongated focal adhesions, cellular adhesions, which is also a marker for fibronectin fibrillogenesis. And I will show that on later experiments. I'll show the fibronectin fibrillogenesis. So whenever you inhibit this glycolytic flux and you inhibit the sugar metabolism, the hexosamine biosynthetic pathway switches from N-glycans and glycolysis to O-glycanylation and oxidative phosphorylation. So that's how this hexosamine biosynthetic pathway acts as a switch, an on and off switch for glycolysis and oxidative phosphorylation. So that's why in the sucrose treatment, whenever you have, due to hyperosmotic stress, you have a decrease in glycolytic flux, so that initiates the hexosamine biosynthetic pathway to, instead of making N-glycans, it makes O glycosylation, and then that's gonna be integrated by galectin 3 and switches all the cellular metabolism to the mitochondria, and that's why you see this enlarged mitochondria, which the mitochondria have their own DNA, so you can see them in the DAPI counter stain as little blue dots everywhere here, and MC stands for mitochondrial content, so you can see all of that here. So then, to further show that this mechanism is working through these branch sugars, I use swaintsonine, which is a Golgi inhibitor. So that's going to inhibit all the glycosylation from being added on to the glycoproteins. So all these branch sugars that you see here will be removed and won't be present in these cells, and you see a drastic change in cell morphology. I also use beta lactose, which is a competitive inhibitor for these branched N-glycans from binding to galectin 3, and you see a change in morphology. And I also use gamma tocotrienols, which shows a similar change in morphology as beta lactose and swaintsonine. So it suggests that gamma tocotrienols are working through a similar mechanism and inhibiting these branch sugars from binding to galectin 3. In control cells, you can see that extracellular galectin 3, shown in green here, is present, and it's present in the sucrose group. However, it does look like in the sucrose group that it's maybe intracellular due to that change in the glycosylation from N-glycosylation to O-glycosylation. And during these galectin 3 inhibitor groups, these 3 treatments, you see a drastic decrease in galectin 3 staining. So can tocotrienol directly bind to galectin 3? I use Schrodinger molecular docking software to dock tocotrienol and beta lactose, shown here and here. And you can see that beta lactose binds the carbohydrate recognition domain, and it binds the carbohydrate recognition domain in a similar way as the chromanol ring of tocotrienol, and both the chromanol ring of tocotrienol and beta lactose both bind the arginine 162 and glutamic acid 184 in the carbohydrate recognition domain. However, gamma tocotrienol can also bind to the hydrophobic binding pocket, which is below the carbohydrate recognition domain. So the Schrodinger docking did confirm that tocotrienols can bind to galectin-3 in a similar way as beta-lactose and other galectin-3 ligands. So does tocotrienol alter galectin-3 expression? I use stain-free Western blotting to show this. You can see the galectin-3 Western blots here. And during a 36-hour 5 micromolar treatment with tocotrienol, if you compare the galectin-3 blots to the total protein loaded in each lane of the gel from the lysate, you can see that during a 36-hour treatment, the galectin-3 expression goes down. So what is galectin-3 localization in these cells? We kind of talked about this before. So these are highly metastatic breast cancer cells, so you would expect the galectin-3 expression to be in the nucleus, which you can see right here with DAPI staining and galectin-3 in green, and outside of the cell shown in A and B. And the way you can look at localization using the confocal microscope is either by utilizing Triton X, very low concentration of Triton X, or not using Triton X. So when you use Triton X, that permeabilizes the membrane. So you can think of it as puncturing little holes in the membrane, allowing the antibodies to go inside the cell. But since the extracellular galectin-3 is bound to the membrane, when you puncture holes in the membrane and you do subsequent washing steps with PBS, you're going to wash away all that extracellular galectin-3, and you're just left with intracellular galectin-3 expression in this treatment with 0.4% Triton X100. So whenever you don't use Triton X, the antibodies that you're using to stain galectin-3 can't go inside of the cell, so you're left with just the staining outside of the cell, which you can see galectin-3 shown here. So for my next experiment, I looked closer. This is just 10x magnification. I did 63x magnification. So can tocotrienol inhibit galectin-3 extracellular morphology? So zooming in with 63x magnification, you can see the little droplet biomolecular condensate morphology that is associated with galectin-3 lattice formation and oligomerization. And during a 5 micromolar treatment with gamma tocotrienols, that gets disrupted. And this is over a 24-hour period. So you can use ImageJ and turn these images to black and white so you can see the morphology a little bit clearer, and you can see that this droplet morphology gets disrupted upon treatment with 5 micromolar of gamma tocotrienol. Can tocotrienol inhibit fibronectin extracellular morphology? So I've already explained that galectin-3 can bind glycosylated integrins and form a dynamic lattice, and that dynamic lattice can also bind to fibronectin and help in fibril formation. And other papers have actually shown that galectin-3 mediates the fibril formation in mammary cancer cells. So you can clearly see the fibril formation in these cells associated with the galectin-3 staining. And in treatment with tocotrienol and beta-lactose, you have a drastic decrease in galectin-3 staining, and you don't have any fibrils forming. So can tocotrienols inhibit intracellular morphology? Because we said that the fibrillar adhesions, so that's the integrins and fibronectin, will stabilize the outside of the membrane and allow the intracellular adhesions, the intracellular focal adhesions, the plaque proteins to bind to the actin cytoskeleton. So you'll have the actin stress fibers forming along with the fibronectin fibrils. So you can see that using vinculin to stain the focal adhesions. You can see them at the ends of the elongated mesenchymal morphology, and that's bound to the red actin cytoskeleton. You also have vinculin within the cell in the cytoplasm because it's constantly being recycled as you have focal adhesion turnover. It's being recycled from these grouped focal adhesions back in the cell so it can be used again. Back deeper into the cytosol, the focal adhesions, this vinculin is still inside the cell. And then upon treatment with gamma-tocotrienol and beta-lactose, you have a disruption in focal adhesion aggregation and a disruption of the F-actin stress fibers being aligned in this elongated morphology. So the elongated morphology associated with the mesenchymal cells gets disrupted. So can tocotrienol inhibit the galectin-3 lattice formation? So now we're just looking at how the galectin-3 protein can bind to itself, so it oligomerizes to form this lattice. So the way you can test for this is the FRET assay, which uses a fluorophore, two different fluorophores, both bound to galectin-3. So you'll have one galectin-3 protein that has a fluorophore that can be excited with 488 nanometer wavelength, and it will emit green light. And then you'll have another fluorophore on galectin-3 on a different galectin-3 than this green fluorophore galectin-3, and it can be excited with 543 nanometers, and it will emit a red light. And you can set up your emission filters to make sure you're only getting the red or you're only getting the green. But then if both of these fluorophores, the 488 nanometer excitation and the 543 nanometer excitation, if they're within 10 nanometers together, then whenever you excite the green fluorophore, it won't emit green light. That excitation will get transferred to the red one if it's close enough, and then that will emit a FRET signal, which is shown right here. So you can actually excite the green fluorophore, and if it's within 10 nanometers of the red fluorophore, you'll excite the green one and get the red signal. And you can set up the emission filters to make sure this is happening. You can see the emission of the FRET signal here, and whenever you use tocotrienol and beta-lactose, it inhibits the FRET signal, which suggests that these treatments are inhibiting the galectin-3 from coming within 10 nanometers of one another, so their oligomerization is being inhibited. So this is in fixed cells with MDA and B231, highly malignant breast cancer cells, but and this is with a confocal microscope, but we also wanted to do it in living cells. So we utilized a plate reader that had incubation. We could had cell incubation capabilities. So we can set the temperature in this plate reader, so we can do the same experiment on the previous slide on living MDA and B231 cells. And you can see that after 1 hour, there was a decrease in FRET signal, but after 6 hours, you had a drastic decrease in FRET signal. So can tocotrienol be used with current treatments? And many papers have shown that it can be used with current treatments, at least in cellular models. However, it can also be used for prevention, and that's where I think it needs to be utilized as fast as possible, not only for the prevention of breast cancer, but the prevention of many other diseases like liver disease, neurological disease, skin disease, in some cases bone disease. So it can be used to prevent a lot of different disease states that are associated with these fibrils and biomolecular condensates, or the activation of fibroblasts. So to continue, I use the most common breast cancer treatment, which is doxorubicin. So doxorubicin works by double-strand breaks in the DNA, and it's good to utilize in experiments because it emits red light. So in the vehicle-treated control group, no doxorubicin was used, so you don't see any doxorubicin inside the nucleus. And galectin-3 here is green in all these groups. So whenever you just use doxorubicin, you can see doxorubicin in the nucleus. And whenever you use tocotrienol and beta-lactose, which got cut off here, you can see that there's an increase in doxorubicin staining inside the nucleus. So that suggests that whenever you have tocotrienol and beta-lactose inhibiting the galectin-3, then that causes an increase in doxorubicin staining. So this is just a preliminary experiment. However, there's been many experiments, as I said, that has shown that tocotrienols can be used alongside many different cancer therapies, and it can be used to prevent cancer. So let's move on to what I think is one of the most interesting parts of this presentation, and that is, does this mechanism provide a framework for emergence? So that led me to publish a chapter in the first edition book titled The Quantum Mechanics of Biological Systems, where I explain that if you think of biomolecular condensates, you can think of them as almost the tiniest bits of emergent patterns in living cells. So normally you think of the central dogma. Normally people talk about, and in school the first thing you're taught is the central dogma of molecular biology, which is that through genetic regulatory pathways, DNA makes RNA, which makes proteins, and then you'll get protein expression. However, this protein expression through biomolecular condensates can form macromolecular structures that end up having an effect on which genes get expressed. So having an effect on DNA, whether it be through methylation or acetylation. So the way that this happens, at least one way that this happens, is when galectin-3 binds to beta-1 integrins through epigenetic regulatory pathways. Galectin-3 can actually regulate itself. So it can turn on its own expression through epigenetics. And that's from it forming this lattice and binding to integrins. However, you can also have the bioelectricity that is playing a role here, because these bioelectric patterns can then mediate if these extracellular condensates are forming or dissolving. So then that adds another layer of computation in these systems and control that isn't really explained by the central dogma. So a way you can think about emergence, which brings me to this figure right here, is you can think about emergence using how the galectin-3 molecule functions through biomolecular condensates and liquid-liquid phase separation. So Plateau's laws is simple, and it just describes how bubbles form and the angles that bubbles form, which can easily be seen in 2 dimensions or a monolayer of bubbles. But whenever you have a multi-layer of bubbles and it's more 3-dimensional, those patterns are harder to see. So that's described by Plateau's laws. And they also describe galectin-3 staining and how you can see galectin-3 condensate morphology in a monolayer of cells here, which you can clearly see these little green droplets everywhere. However, when you have a multi-layer of cells, that droplet morphology isn't really visible anymore. So the reason that this happens is you can think of all these little droplets as being grouped together and kind of forming a substrate on top of the cells, and that substrate allows for the cells to migrate upwards. And this is in highly malignant breast cancer cells, and the thing about cancer cells is they don't have any contact inhibition. So whenever they grow and expand the whole surface that they're growing in, for instance, like a dish. So once they fill the whole dish and they don't have any more room to grow, they don't have contact inhibition like normal cells. So then they'll just start growing on top of each other. And so they'll grow in a multi-layer. And you can see that where this multi-layer is occurring, where this multi-layer of cells are occurring, the top layer is this galectin-3 staining in green right here. And I proved that this is a multi-layer of cells using depth coding. So this is using Z-stacking with the confocal microscope, which you take many different images of every layer of cells in this specimen right here, and then you stack them together to generate a three-dimensional image, and the depth is shown in colors right here, where the topmost portion of the specimen is red and the bottommost portion is blue. So you can see that everywhere is that stained green right here, you can see it as green, the green portion, which is the galectin-3. So the galectin-3 biomolecular condensate, the lattice formation of galectin-3 forms a scaffolding that allows these cells to start growing on top of each other and forming a multi-layer of cells. So this brings me to my final figure in this paper, titled Biological Complex Adaptive System with Multi-Scale Computation. So I'll bring up water again, and you can think of how water can form these low density and high density structures, and how you can compare this to prime numbers, and how even prime numbers, which is just math, even prime numbers can have this pattern, forms this pattern within math. And the pattern that it forms has been compared to quasi-crystal structure. So a quasi-crystal is, think about like a salt crystal is a repeating uh, pattern of sodium chloride, just constantly repeating in a lattice. But then whenever you have a quasi-crystal, that's a pattern that is similar, self-similar, but it never repeats, kind of like a fractal, because a fractal is self-similar, but some of them never truly repeat. They just look very similar. So that kind of gives you a conceptual idea in your brain of how these prime numbers, how they form patterns even within math. So in the same sense, if you can have these hidden structures in math that form these patterns that aren't exactly repeating but still patterns, then why couldn't water and why couldn't amino acids and nucleic acids, why couldn't these molecules be forming these same patterns? And I think that they are. And I think they are together through liquid-liquid phase separation, so that you can, and they all work together to form these patterns, which enable them to form this emergent structure. And I think that it can happen in the case of photosynthesis. These patterns can happen all the way to the particle scales, all the way to the femtometer scales. And all of this together through liquid-liquid phase separation and biomolecular condensates, those are the tiniest emergent patterns that are forming, which organize cellular morphology and generate all the different morphological patterns we see in tissues today, which is shown right here, which surprisingly has some of the same patterns that we would expect to see in the low density structure of water. And that's why whenever I made this figure, I made sure to have a blue background, because the water is the underlying scaffolding for biology, at least on Earth. So how can we test this mechanism? So we can test this mechanism, as I mentioned before, through bioelectricity, and using bioelectricity to modulate the liquid-liquid phase separation. So as I said before, different experiments have shown that ions can regulate liquid-liquid phase separation. And right here, I used a voltage-sensitive dye. So the more green dye that you see, the more depolarized the cell is. So one way you can think about this happening, if tocotrienols are inhibiting the biomolecular condensate formation outside the cell, then that means that the condensates were holding the ion channels, because ion channels can have in glycans. So whenever you have the biomolecular condensates holding and stabilizing the ion channels outside of the cell, then that is enabling the polarization of an electrical field. But then whenever you disrupt the condensate formation, you have a disruption in the localization of the ion channels. Some of them might get degraded and get put back inside the cell. So you'll have a disruption in this electrical potential, and that's why you see the cells as depolarized. And I also used amiodarone, which is a commonly used as an ion channel inhibitor, as a control, just to show the depolarization of cells. So another way you can do this is by using ions like manganese to induce the liquid-liquid phase separation. So normally you have, in these pictures, green is galectin-3. So normally you have a monolayer of cells which express galectin-3. And then if you add manganese, which will induce the biomolecular condensates to form. So just like in my previous experiments, whenever you have that galectin-3 condensate that forms, that renders a scaffolding for the cells to start proliferating on the z-axis and proliferating upwards. And what you can see here is during the manganese treatment, you see little spheroids that are forming everywhere. So normally you just have, in growth media, you just have a monolayer of cells at this point, and you can see the monolayer of cells have an even distribution of galectin-3. And then whenever you add manganese, you induce the spheroid formation everywhere, which also are completely surrounded with galectin-3, which is stained in green right here. So you can see in this confocal Z-stack. I'm going through this spheroid specimen, and you can see that the whole thing expresses galectin-3 everywhere. So it seems like galectin-3 is acting as a scaffolding for this multi-layer cell growth. And this image right here just shows the bottom and top of the spheroid along with the middle of the spheroid in green. So the bottom's in blue, the top's in red, middle's in green. And you can get an idea of, and these are just preliminary experiments, but this gives you an idea of how you can either inhibit the biomolecular condensates using galectin inhibitors, or you can induce them using manganese and possibly other ions like sodium chloride. You can induce the biomolecular condensates to form, which will enable the cells to behave in a different way and have different morphologies like multi-layer growth or spheroid growth. So these are just preliminary experiments, but it does show that whenever you modulate these biomolecular condensates, you have a change in cellular morphology and a change in bioelectricity. So the take home of all of this, there's a lot more take home than this, but I think this is the most interesting take home, is that understanding the hidden mechanisms behind emergence represents the beginning of a new science. So understanding this will help us hopefully understand how to prevent certain diseases like cancer by modulating these biomolecular condensates. I think it will also help prevent certain things like liver disease, where fibrosis is a big part of the progression of the disease. It will also help with things like Alzheimer's, where these fibrous plaques play a big role. But it will also help us understand biological organization and how these emergent patterns you see in biology isn't random at all. You just have to understand every agent that is working together to form these patterns. So whether it be the different density states of water, whether it be the electrical fields, whether it be the DNA, RNA, or proteins associated with that condensate, or the post-translational modifications of those agents and how they can change the condensate. This just opens up a whole new science, which is combining, I think, many different fields of science. So to understand this new type of science, you're not going to be able to have boundary conditions like chemistry, physics, and biology and math. You're going to have to think of it all as one subject and one science. So I hope you enjoyed this and you have a wonderful day. Don't forget to like and subscribe.